more myelin. It's like a wire that has plastic around it that makes it run more efficiently and more effectively. We develop myelin in adolescence. And cells that are used more frequently have a better lubrication of the axon itself to provide a stronger and more efficient signal between the axon, between neurons. So I want to say a couple things about these cells. These are so tiny. You can fit 50,000 neurons on the head of a pin. Yeah, it's amazing. And it took us a long time to realize that they don't actually touch because they're so small. Once we developed the microscopes to see, there's little clefts, little what we call synapses, in between, these little gaps in between these cells, and that's where the chemicals are. And when we say someone has a chemical imbalance, we're talking about neurotransmitters, which are chemicals in between these cells that affect how they communicate to each other. 50,000 of these can fit on the head of a pin. There are 100 billion inside your brain, which is amazing in and of itself. But each neuron has around 10,000 connections. So imagine 10,000 connections to this little itty bitty cell. Imagine if you were having 10,000 conversations all at once. It would be so much information. But these cells can sort through it. And when these connections inform the cell that there's enough stimulation, there's enough electrical energy, it gets to a certain point, and once the electrical energy gets to a certain point, it fires. It's like a gun that when you pull the trigger a little bit, it gets to a point and then it just fires. And that whole metaphor of the gun is appropriate because these cells either fire or they don't fire. There's no halfway. The way in which the brain is able to understand intensity or pressure is that there's more neurons firing if there's more pressure, like on your skin or something like that. And there's just less neurons firing if there's less pressure. The firing is always the same amount of intensity, just pew, pew. But 100 billion cells in the brain, 10,000 connections. I'm pointing to the board because I like to write this. Yeah. 100 billion times 10,000. If I write it on the board, you got to write it in your notes, right? 10,000 connections. And again, these are 100 billion cells in the brain. If you multiply 100 billion times 10,000...
Quadrillion.
Yes, who said that?
Ridiculous. I just looked it up because I was interested.
Yes. You guys are awesome.
That's a whole lot.
1 quadrillion is, quote, ridiculous. That is true. Luke, that was you, right?
Yeah.
1 quadrillion connections in the human brain. Here's what gets literally ridiculous. There's a quadrillion connections. They're like switches, like light switches that can be on or off. Now, you have a quadrillion switches in your own brain. There's 14 quadrillion in this room, right? That's amazing. But just in your brain, there's a quadrillion switches. These switches can have different combinations and different possibilities. Some switches on, some off, some on, some off. And so the variability, the number of patterns within your brain based on these 1 quadrillion switches, because it could be so many different ways, is more than the atoms in the visible universe. The capacity of your brain is essentially infinite, which is mind-blowing, right? I mean, your brain can't even understand itself in terms of its capacity. Pretty cool. Good. I'm glad that you guys are really appreciating that. I think that's amazing. Let's turn now to a little video, a little animated video of what this looks like. You can see here in the video an action potential and the chemicals coming to the synapse. sodium and potassium ions going in and out of the cell, creating that electrical current through the axon. It's positive on the outside, negative on the inside. Pushes these synaptic vesicles to the terminal button here. And these little red things are the neurotransmitters. They fit like keys into holes, almost like an outlet into a plug that opens up channels for the ions to flow, the electrical ions to flow to the other neuron, to the receiving neuron. And then if enough stimulation happens, it will fire. Sometimes the neurotransmitters are communicating not to fire. Sometimes they're saying to fire. That's how they communicate with each other. Any questions on that? Any questions on how that works? So those chemicals in between the cells are absolutely key. They have a very important role in the function of the mind and the nervous system, our thoughts, our memories. They affect how neurons communicate to one another in pretty substantial ways. You should write this down. Neurons that fire together, wire together. It's a nice, catchy phrase that conveys how when parts of your brain are active and then they connect by making these, they got 10,000 connections, they kind of can wire together. And in fact, long-term potentiation refers to how neurons change and they essentially become wired together after something has been learned so many times. We've learned this with sea slugs because they have huge neurons that you can zoom in and you can see how these neurons actually change as the sea slugs learn that a wave is coming and they get splashed a bunch of times and these neurons change. And so right now, your neurons are changing. And the way in which they're changing is there's more synapses, perhaps. Maybe some neurons, based on what you're thinking about, how your neurons are firing right now, are kind of developing more of these little connections. And if you do it enough and you keep ingraining this into your mind as you study, your brain can permanently be altered so that these you have what's called long-term potentiation, which is a substantial increase in serotonin, which is a neurotransmitter that basically balances a lot of things. It has to do with mood and other things, but increases in serotonin and more synapses when your memories are essentially become long-term memories. Okay. What is going on in this slide is it's talking about reuptake. So when these neurotransmitters are in the gap here, the synapse, the synaptic gap, you can see it right here, synaptic gap, three things basically happen. They're either absorbed here and the receiving neuron kind of breaks it down. It can be broken down by enzymes in the synapse, or it can be reabsorbed here. So there's basically three things that happen. One way that a kind of antidepressant works is it blocks reuptake. So it's not reabsorbed here. So that it's more likely to be absorbed here by the receiving neuron. And that's called an SSRI. Selective Serotonin Reuptake Inhibitor, because it inhibits reuptake. So it's not reabsorbed into this earlier cell, and it's more likely to reach those receptor sites and cause more firing in that receiving neuron. And serotonin has a way of balancing things. It kind of affects other neurotransmitters. It definitely has to do with our mood. And so you can see that here. Serotonin is the first neurotransmitter listed. It affects our mood, hunger, sleep, and arousal. And more serotonin is generally good. It's also used, SSRIs are used for when individuals have kind of obsessive thoughts, maybe obsessive compulsive disorder, because serotonin kind of has a way of balancing things out a little bit. Okay, so let's talk about these neurotransmitters. I know we've said a lot of key terms here, but one thing I could ask you about are some of these neurotransmitters, like acetylcholine that's on the board, glutamate, things like that. So serotonin has to do with mood and so forth. Dopamine, I like to think about how dopamine is often the case when it comes to rewards in the brain. When you're reaching a high, an emotional high in life, or you're some kind of, the reason is because dopamine is definitely affected by drugs like cocaine, methamphetamine actually mimics the dopamine molecule very well, which is why, why it's so addictive. But it also explains why there's a really serious side effect with hallucinations and psychosis when you're doing methamphetamines, because it's a lot like schizophrenia in terms of how it affects the brain. Acetylcholine, I like to think about, it definitely has to do with Alzheimer's and memory, but I always remember like when you get muscle soreness, it's because of, it's partly because of how there's acetylcholine and other chemicals. When you get bit by a spider that's venomous, it affects the nervous system so that there's a tremendous release of acetylcholine. It's almost like the spider is releasing acetylcholine into your nervous system, causing muscles to contract and eventually seize up. And it's just amazing how that works. It freaks you out, yeah. Norepinephrine has to do with arousal and energy and alertness. This is affected by exercise. You get an increase in norepinephrine. Epinephrine is similar to norepinephrine, and it has to do with arousal as well. GABA is a major inhibitory neurotransmitter. It tells other neurons, do not fire. It's a nice balancer when people are having too much thinking or they're kind of anxious, and so some anti-anxiety meds affect GABA, and they basically calm you, they kind of relax you, and a lot of the thinking, if it gets out of control, can be kind of balanced by increases of GABA within the brain. One of the things I think is interesting here is, guess what else affects GABA? Alcohol. So when you're drinking alcohol, you have an increase in GABA, and your things are slowing down, you're kind of, it's a depressant, so it's decreasing the central nervous system activity, the firing of your brain goes down, which is why eventually you die if you drink so much alcohol. If you give blood alcohol level that's high enough. But this is the part of the brain that we're turning off, and it basically says, don't do that. You may have an impulse to do something, but there's part of the brain that says, you might not want to do that. But when you're drinking, that part of the brain is not firing, and it's like, that's why people dance on the table. It's because that part of the brain is basically offline and is not there to tell you, don't do that. And so you have less ability for that part of it. Basically, the prefrontal cortex to say, regulate impulse control things. Yeah.
Is that why it's like also like alcohol doesn't make you do things that you wouldn't already do? It just lets you do things that you already wanted to do, basically?
Yeah, it cuts off the part of the brain that does a lot of the regulating and self-monitoring and all that kind of stuff. Basically the frontal cortex. So there's this sort of more impulsive aspect. We have impulses, but we're not really checking that and we're not really governing. So, sort of, yes. Glutamate is a major excitatory neurotransmitter, which is involved in memory, but it's firing neurons. It's involved right now when you're thinking about new things and so forth. And then endorphins, this comes from two words. Dr. Skolsky's breaking up words again. Endogenous morphine. We knew about morphine. We knew about morphine before we knew about endorphins. We found that morphine would bind to receptor sites in the body. And we're like, why are there these receptor sites? There's got to be a natural morphine in the body. If morphine's going to bind to something, there's got to be a reason that they have these keyholes. There's got to be keys somewhere. And then we discovered there are keys, and we labeled them natural morphine. And it's pretty amazing what it does in terms of pain and pleasure. Okay, we've got a few minutes left. This is the fun part. Take out a piece of paper, and just a separate piece of paper, write down one of these neurotransmitters very large on this piece of paper. and then crumble it up into a ball.
Really big?
What's that thing?
Really big?
Really big, yeah. I mean, yeah, big enough so you can read it. Yeah, you can stop. I guess you can stop.